how do you see the next generation nucleic acid delivery system fitting into the structure of what you want to be working on and making the change that you want to see?
Sure, yeah. So in my academic career, as I said, I'm focused on what makes a cancer cell that's likely to spread different from one that's likely to stay put. And so we've developed actually in the chick embryo model ways of doing unbiased screening to discover new genes that are driving that spread of cancer. And we've been pretty successful. We've identified over 20 so far. All of these are completely novel, not described to affect metastasis previously. And for at least 6 of them, we've been able to demonstrate if we hit just that one gene, we can completely block metastasis, by over 99%. So the goal for sort of developing this innovative genetic medicine technology, it's called the fusogenics proteolipid vehicle. So it's similar. We think of it as sort of an lipid nanoparticle next generation. The goal for that in my lab was to use that platform to cure metastatic cancer using these gene targets that we discovered in my research. And we were looking at sort of the other gene delivery mechanisms that were available in the market. We all know about LNP or lipid nanoparticle now because we've probably taken Moderna or Pfizer's vaccine for COVID. You may be familiar with the gene therapy platforms, adeno-associated virus or AAV. This now has a commercial product for eye diseases, for instance, for gene therapy. And in 2016, we realized that neither of these technologies were really going to be able to blow open genetic medicine, be used for CRISPR, be used for RNA vaccines, be used for all of these amazing tools that are being developed. And so we decided, and we saw that our fusogenics PLV technology, we could deliver DNA, we could deliver RNA, we could deliver gene editing. And we thought, we have the opportunity to go after many diseases other than cancer. And so we launched Antos Pharmaceuticals in 2016 with exactly this purpose: build a genetic medicines platform technology company that would begin to develop tools to be able to do gene editing, gene knockdown, gene therapies, and without another technology that was able to do all of these things, we saw a huge opportunity for us to cure a lot of diseases that would be otherwise incurable.
Do you feel this is something I talk with my friends about all the time, which is, how do you take something negative and see, the positive of it? And I felt that I feel like sometimes when we talk about COVID, it feels like kind of like a Manhattan Project level thing happened in terms of like so much money went into technology similar to this. And I'm always wondering, what are some of the positive, ramifications of this? Did COVID accelerate anything you were working on in terms of, bringing it either closer to market, making it easier for people to understand? When you're talking to an investor, depending on your investor, a lot of them aren't really all that technical. They have like technical people to help them. So maybe like they're more familiar with the concepts. They're more willing to fund you, which, has this proliferating effect from there. But if we look at COVID as like a Manhattan Project level thing that proliferates, nuclear energy and, reduce the cost of energy and that type of thing, what benefits do you think your company, what you're working on has generated from it?
Again, COVID had a dramatic impact on us.
Yeah.
At the beginning of before the pandemic, as we were just sort of hearing the news about this virus and, overseas, we were really bringing this fusogenics PLV technology. Our first in-human trial was going to be in oncology. And we had sort of identified the principal investigator. We'd sort of thought about manufacturing. We're getting ready. We've designed the trial. We're all ready to go. And then, COVID hits, lockdown happens. We worked pretty extensively at the University of Alberta as well. And the university, was planning to shut down all the research labs. And we were familiar with Moderna. We're familiar with BioNTech and Pfizer. And we had a pretty strong feeling that the RNA LNP technology would be successful as a vaccine. But we also knew that it would have pretty key limitations in the fact, basically, that RNA is, it's designed to be labile, it's designed to degrade quickly. And so we thought, the freezing to -80 and the cold chain for RNA vaccines would be a challenge, which we've seen. But and it's really created this vaccine inequity across the world where we have access in first world countries to these vaccines and not in third world countries. And we also saw that RNA would not likely be as durable. And we've seen that as well. So in teenagers, for instance, some of these RNA vaccines have durability in weeks instead of months. And even for adults, maybe less than 6 months. And we thought, because our platform is similar enough to LNP, but we can deliver DNA. DNA is super stable, it's very simple and inexpensive to produce. And it should be much, much more durable than RNA. We decided to pivot from cancer and develop a DNA-based COVID-19 vaccine. And so that completely changed, obviously, the direction of the company. We were very fortunate to get support from the Canadian government for that program. And we were able to take this platform that at the time was sort of a research and development platform. So we had a few partnerships at the time, but we hadn't gone to clinic yet. And now we've just completed a phase 2 clinical trial with our COVID-19 DNA vaccine. We're looking forward to coming back to North America to do phase 3 as a booster. And it really forced us in a very sort of high stakes, high energy environment to solve, manufacturing, to solve regulatory, to really mature the platform to a point that we're able to get, we've now had exposure in 268 individuals who received the vaccine, very clean safety. We're very happy about the way we manufactured it and really opened the door for not only other vaccines, but for other gene therapies.
One benefit that I could see with it, mRNA not lasting as long, is that they talked about, at least I remember this, it was a couple years ago, but they talk about like if we could just get everyone to stop moving for 2 weeks, it would kill the illness because the spread rate and stuff. Like that's why everyone like kept trying to do lockdown. So if you can improve that for a short amount of time and then it goes away, that's kind of nice. I think one of the concerns that people had is that how long. I know a lot of people had this because I had an episode on the COVID vaccine and it got some great comments there. But people were concerned about the long-term effects of it. So so like the mRNA kind of like trailing off is kind of a nice feature. Does the DNA version of your vaccine have alternative complications where you would see something like not for years, or like how would like the converse of something that is more stable affect stuff like that?
That's a good question, and it's something that we thought about and looked at as well. I think when it comes to vaccines, though, I mean, I guess there's two things. So with a vaccine, you're basically think of a vaccine as basically a wanted poster, for the virus. You're showing your immune system the wanted poster. Hey, you see this guy in the future, mount a response because he's bad. And but it's only a poster. So you're not really introducing anything in and of itself that's dangerous. It's a very, very tiny amount. You're basically showing a picture of the virus. And so, and there's really other aspects to it too. Our platform, when you inject it in the muscle and you make a little bit of the coronavirus protein or whatever, virus that you're trying to protect against, it's just made in your muscle. It doesn't go anywhere else. And I think, and that's in contrast somewhat to some of the other platforms that can be found other places. And so we obviously talked to Health Canada about it as well. And we perceive the risk to be pretty much zero, and which has been borne out by our clinical studies as well. I would say one of the issues of durability is really important, in preventing viral transmission, because you want very high titers or very high levels of those neutralizing antibodies for as long a time as possible, because, they are the things that prevent the spread. And so if your protection falls off in a couple months, then you need another booster to get that protection.